Hello.
Hello.
Brad.
Hello.
Maybe we're about ready. What do you think, Anita? We all here?
Yeah, I think go ahead and get started. People might trickle in, but yeah, go ahead. That way you have enough time.
Okay, great. All right. Good afternoon, everybody. Welcome to our first journal club of this year, right? Our first journal club session. So I'll just explain a little bit about how we'll run these journal club sessions for the fellows. So what we'll do is when it's your turn to do a journal club, you will liaise with one of the faculty. Typically, it's me or Galit Dunitz, and we will do the faculty will do about a 15-minute brief kind of review, if you like, of some statistical issues or statistics, just because we recognize that many of you might not stay in academic medicine. You might go into private practice after you've been here. So we just want to make sure that you understand how to critique journal articles, what to look for, think about methods and some basic statistics, since this might be the last chance that you get to go through some statistics. So we will take, like I said, about 15 minutes of each of the journal club sessions to discuss some of these, and then we'll hand it over to the fellow who will be presenting the journal club article. Okay, so I will do the first one. This is kind of it's not really statistics, it's kind of a pre a pre rec to thinking about research for those of you that might want to go on and do some research. So I am going to share my screen. I hope you can all see that. And I'm just going to turn the cameras down so I can see what I'm looking at. So if you have any questions, just feel free to jump in. But we'll start to talk about hands-on hypothesis formulation. So if you're going to do research yourself, or if you're, reviewing articles, this is the most important thing that you can do to start is think about your hypothesis, because if you want to do a research project, you can't just go, oh, I'm just going to go and pull these medical records and look at X and, see what it's related to. It doesn't work that way. There's a whole, way of doing this to do it properly. So, as you know, clinicians look to literature to guide your clinical practice, which we know is evidence-based medicine. And really, it's the responsibility of the people that do the research and to attempt to answer questions that are well thought out, clinically relevant. And, when you develop your research question, you have to have an hypothesis and, usually your objectives. Okay. So, there are challenges. This is not an easy thing to do, right? There are challenges to developing an appropriate question. It takes a lot of back and forth with your collaborators because, first of all, you have to think about, which things could or even should be studied, right? And you have to rationalize the need for doing this investigation. So I think you all know that, there are several ways you can increase your knowledge about something. You can do a literature search, you can look at a systematic review, you can have focus groups, or it could even be, your patients that have brought questions to you that you're like, oh, well, I don't think anybody's done that. Why can't we look at that? You can talk to experts. You can just be aware of advances in the field. I think the technology advance is kind of pretty pertinent in this field these days. And you can also do your own pilot data collection. But really, all research projects share the same general structure, which looks a bit like an hourglass, right? So you can see that here, this hourglass, where you begin with this broad set of questions, quite big. You narrow down something to focus on, then you operationalize it. And then you can start to, you have your question, you observe, you collect your data, you analyze your data, you reach your conclusions, and then you can generalize it back to potentially, various populations. So just think about it as this hourglass idea. And so in these boxes here, box number 1 is the FINER criteria. These are criteria for a good research question. Make that a little bit bigger so you can see it maybe. There we go. Okay, so FINER stands for feasible, right? So first of all, is what you want to do feasible? Can you?
Can I ask you a question?
Yep, you can.
I don't know if I'm the only one. I probably haven't had enough coffee this morning. But the hourglass,
Yes.
I'm not missing the point on the hourglass. Is it to say you start general somehow and you get very specific in the observations and then you end up generalizing to
Because I think people start with really big questions, right? And it's the discussions and the back and forth to basically hone in your idea. So you really have to bring it from this big idea that you have down to a question that can be answered.
which is very specific, and that's what's narrow, you're saying.
That's your specific aims essentially is your narrow bit, right?
And
then
then
you can generalize it.
I see. And then all the way down to the last paragraph of the discussion on the paper that you write, where you're
Yes.
allowed to speculate and look
Yes.
at implications. I understand.
Yeah. Okay, so is it feasible, right? Can you recruit enough people? Do you have the expertise? Do you have the money, right? That's a big one. Can you manage to do it? Is it interesting? Okay, if it's, I mean, some things aren't so interesting to all of us, but is this a topic of interest to the experts in the field or to the community, the patient population, etc. Is it novel, right? Does it confirm findings? Does it refute findings? Does it extend findings? Is it new findings, right? Is it ethical? That's an important one. You can't do it if it's not ethical. It has to be approved by an institutional review board. Or is it relevant, right? There are lots of things you could do that might have no relevance to science, no relevance to health and policy or to future research. So it has to be something relevant. So think of the FINER criteria when you're thinking of your research question. The next box is the PICO criteria. So I think we've all heard this one before. So the P is your population. Who's your population? Who you recruiting for this study? The I is an intervention if you have an intervention study. The C is your comparison or your control group. You're always your outcome of interest. And then T, which doesn't happen in all studies, but T is the element of time if you're doing longitudinal studies. So you can think that, if you don't put enough effort into your box 1 or your box 2, and you don't have the resources to do it properly, the quality of the study and obviously its results will be compromised. So I think you can, see those parallels to clinical care, right? If you don't put in the effort and, be as knowledgeable as you can about your field, then that does have an impact on clinical care. So it's a similar issue when we come to doing research. So a hypothesis, what is a hypothesis? It is a tentative prediction about the nature of a relationship between 2 or more variables. So can anybody tell me what else you could say about a hypothesis? There's a couple of things that a hypothesis has to have or has to be a hypothesis. Any ideas? Has to be testable, right? Hypothesis can be tested. You have to test it. A hypothesis cannot be neither too specific nor too general. It's essentially a prediction of consequences, right? So if I do X, then Y happens. Hypotheses are not moral or ethical questions. And a hypothesis is valuable even if it's false. Okay. So I'll give you this example I'll read the example, then I'll let you read the options at the bottom. So you are a nutritionist working in a zoo. One of your responsibilities is to develop a menu plan for a group of monkeys. In order to get all the vitamins they need, they have to be given fresh leaves as part of their diet. So choices you consider include leaves of the following species A, B, C, D, and E. So you know that in the wild, these monkeys eat mainly bee leaves, but you suspect it could be because they're safe while they're in the bee trees, whereas eating any of the others would make them vulnerable to predation. So you are going to design an experiment to find out which type of leaf the monkeys actually like the best. So you offer them all 5 types of leaves in equal quantities, and you observe what they eat. And so we have an A, B, C, D, E for potential hypotheses. So which one of these could be a correct hypothesis? Any willing ideas? Anybody wants to hazard a?
I don't see any hands up,
do you
but go ahead and
mind,
unmute.
Okay, I can't see, so just go ahead and shout because I can't see the camera.
do you mind scrolling back up to the question stem
Sure,
just
yeah.
briefly?
Yep. Yeah, you're either going to have to shout out or somebody's going to have to tell me about hands because I can't see
We-
the screen with all of you.
We think B or C, but
Oh, well, you were right. It is B. Why do you think it's B?
it's the if then, right? That's the hypothesis. If offered 5 types of leaves, then the monkeys will eat on B leaves.
Right, so it's prediction and it's testable, right? Those are the 2 criteria for hypotheses. It's a prediction, so it predicts the outcome of the experiment and you can test it, right? Because you can do the observations and then you can see how many leaves they ate, right? So types of hypotheses. Null hypotheses, I'm sure you've all heard of that, and the alternate hypothesis. So the null is the hypothesis of no difference. The alternate hypothesis is the opposite of the null, reached if the null is rejected. And often it's the alternative one that we're trying to support, right? Because we usually want to test if, let's say, drug A is better than drug B, right? So here's some examples. So in a clinical trial of a new drug, the null hypothesis would be that the new drug is no better, on average, than the current drug. And so you would write the HO, the null hypothesis, there is no difference between the 2 drugs. The alternate hypothesis might be that the new drug is different on average compared to the current drug. And so you would write the H1, the alternative hypothesis, the 2 drugs have different effects. Or you could write that the new drug is better than the current drug. So you could write the alternate hypothesis, the new drug is better than the current drug. Okay, and usually your final conclusion is given in terms of the null. So you either reject the null hypothesis in favor of the alternate, or you do not reject the null. Okay, but we never actually conclude that we reject the alternate or accept the alternate. It's usually done always with the terms of the null hypothesis. So that is all I have for you. Like I said, this was the quick one. This doesn't really involve stats, so I will stop sharing so I can see all your faces again. Any questions now that I can see you all? No? All right, well, we can hand it over to Journal Club and Brad is going to take the lead.
Thank you so much, Dr. O'Brien. My name is Brad, as you guys know. Hey guys. I am happy to present to you today the first Journal Club of the year. So, without further ado, let me share my screen. Okay. Oh, jumping ahead. So the article that I had the pleasure of reading is called Fitbit Measured Sleep Duration in Young Adolescents is Associated with Functional Connectivity and Attentional Executive Control, Memory, and Sensory Networks. But before we begin, I would like to take you all, hold on. I'm gonna have to reshare my screen here. Make sure you guys can hear. We're gonna do. How do I get rid of this?
Then you put the bar beside.
Oh, okay. Well, hold on. Hold on. This is more for me, to center myself. So
And thanks for gifting yourself these next few minutes.
please
It's important
relax.
to remember that you're a
And let's do a
priority,
5-minute
and
guided
allowing yourself even just a few short minutes of intentional reflection can really have a positive impact on the rest of your day. So let's use these next few minutes to come back to center and set an intention for the rest of your day. Move into a comfortable position, whether you're seated at your desk at work or laying on the couch at home, and gently close your eyes and shift all of your attention onto your breathing, taking slower, deeper breaths than you've taken all day so far. Taking a deep breath in through your nose and slowly letting it out through your mouth. And continuing to breathe that way, feeling your lungs expand out as you inhale and contract back in as you exhale. Tune into your body, noticing how it feels. Noticing if there's anything that it's trying to tell you. Notice any place of tension or tightness in your body. And give those areas permission to relax, sending love into those areas. Thank your body for taking such good care of you, and let it know that it's okay to rest and relax for these next few minutes. You may notice that your mind starts to wander off. That's okay. That's natural. Just notice it and bring your attention back to your body, using your breath as your anchor. Try to picture one thing that's happened today that's made you smile, or made you thankful or appreciative. And just let that feeling fill you up for a moment. Breathe that feeling in from the top of your head to the tips of your toes. And allow yourself to smile if that feels natural. Now focus on something that you can do today, whether it's for yourself or for someone else, that will allow you to continue feeling this way. It could be something as small as deciding to go to bed early tonight, so that you have time to finally read that book that's been on your bedside table. Or the joy of getting dinner with a close friend. What's one little thing you can plan for your day to bring that intentional joy to it? Now just focus on that for a few minutes. Sit in that feeling of joy and peace. Now let's take a few more deep breaths together. A deep breath in, feeling your lungs expand out as you inhale, and everything contract back in as you exhale. Again, an even deeper breath than the one before it, holding that breath for a beat, and exhaling everything out. Last time, your deepest breath yet, sending that breath through your entire body. And exhale it out. And in your own time, slowly bring your awareness back to where you are and thank yourself for taking these few minutes to just be intentional and kind to yourself. And when you're ready, you can gently open your eyes and enjoy the rest of your day.
Thank you so much. If you get anything out of this journal club, just remember that it's okay to take 5 minutes for yourself, okay? All right. So the objectives for today's talk is we're going to talk about what this article brings, what the study adds to the body of literature that we already have. We're going to critically appraise the design and interpretation, discussing the weaknesses and the strengths of the article. And then we're going to discuss the implications for practice and future research and how the article may have impacted your clinical practice. So I started with the PICO, and so trying to make this as interactive as possible. Please, feel free to speak up and chime in. But so we'll start with the P, the population. The study was took 37, almost 3,800 adolescents, ages 10 to 13 years old, which is part of that ABCD cohort, which was this large national multi-center trial of initially doing sleep logs and parental surveys of how much sleep they thought their kids were getting. And then after about a year or two, they switched to the Fitbit, and then, of course, over 2 weeks, and then looked at fMRI studies of their brains. So does anyone want to speak up about the population? Like, what do you think of this number for a pediatric research publication?
I don't know, I think developmentally there's a big difference between a 10 and a 13-year-old. I mean, I think of the patients I would see in clinic, and I mean, I know it said young adolescents, but I don't know, like a 10-year-old boy and like a 13-year-old boy could be very different. So I'm interested about that.
Yeah, no, that's a great point. And I think that, they did try to, for girls at least, right? They talk about this later where they looked at premenstrual and then after menstruation. So they do differentiate that, but yes, I agree. But what about the number, just the number? I mean, for me, for a pediatric research publication, that's huge, right? I'm so used to pediatric studies being like 20 kids, you know what I mean? So I think, in terms of the strength, this is definitely one of the strengths of the paper is very robust, right?
So the sample size is large indeed, but it also depends with what is the outcome, right? Because if you're looking for rare disease outcomes, then and you have 20 cases, then it's not a large sample size. It's all in the context of the outcome.
Right. Okay. And then the exposure, it was this 2 weeks Fitbit measured sleep is what the study looked at, right? Does anyone want to talk about the 2 weeks of sleep, what they think about that, a large part of the study was based on this 2 weeks of sleep? Or, I guess it's probably the best with what the big had could work with.
I think 2 weeks is good. I mean, I think a lot of the time that's what we look at as well, right? Like when we're doing MSLTs, we look at a 2-week window, and so you maybe decrease the chance of day-to-day variability. But again, depends on when this 2 weeks is. Is this during school time? Is this holidays? All of that. So that would be something interesting.
Yeah.
I think that also even trying to do more would be a little more cumbersome for an adolescent
Sure.
to keep it on
Yeah.
and keep a log and be consistent. So I feel like it's fair.
Yeah. And I thought since with like such a large study cohort population that would kind of hopefully minimize any variability, and like what you guys were talking about in like time of year this was taken, and go ahead, Dr. Chervin.
Yeah, but the other thing I think to remember is that it's actigraphy, so it doesn't measure sleep. It gives you an estimate or approximation of what sleep is doing in that time.
Yeah, and I think the article goes into how they only looked at sleep duration, like they did not break it down into like, you know, deep sleep, light sleep, or REM, which we know that this Fitbit has the capability of doing. And so yes, and also I don't know if anyone saw, but it says 2 weeks Fitbit measured, yeah, sleep duration, but they had a minimum requirement of 5 days, right? So if you had less than 5 days of sleep, they excluded you. So I mean, in that 5 days of sleep, right, that they may have included in this study, you know, think about if two to three of those nights are on a weekend versus the weekday, right? That may vary significantly for each child, the amount of total sleep that they're getting. And then the comparator, this is a cross-sectional study, right? So we're not really comparing, it's just looking at different sleep durations across different age groups. And then our outcome is looking at resting state fMRI connectivity. And so now I just want to talk about the methods a little bit. Oh, so full disclosure, I used ChatGPT to make these slides for me. We don't have to do this. Activity, delete that. Okay. So these are some potential biases. Like I said, the cross-sectional design, right? It's a snapshot in time. It's looking at this already published set of data. Fitbit, right? We kind of already talked about the, you know, Dr. Goldstein, I think, had a great lecture about the limitations of wearables versus like the gold standard of polysomnography. And one thing I noticed in the study is that, you know, I don't think they did like screening for sleep apnea in these kids, right? So that could be like a big confounding factor in the population, is if they had, you know, underlying sleep disordered breathing affecting the results of the study. Selection bias, which I'll go ahead, I'll pull up the chart here so that we can kind of talk about this in more detail. So let me see if I can find the article. Okay. So just looking at this right here, this infographic right here, can someone tell me what this is or what they think of these numbers?
I mean, the number of boys and the number of girls by the end of all the exclusions were pretty equal. So I think not a huge difference.
Yeah. And I believe this initial N, the 11,876, this was like the ABCD data where they included like the data logs and you know, earlier studies. So and you know, it was across 21 different centers. And so one of the things is that they mentioned is that they tried to represent the demographics in their area of each of these places that, you know, standardizes the American population. And so as you can see though, you start with this about nearly 12,000, and then we end up with 4,000. So about, you know, a third of the patients drop off, which is a pretty significant number. And then, you know, they're excluded for Fitbit data, or missing Fitbit data, missing fMRI data, right? So that's a big one. Yes, Evie.
What is the excluded outliers for fMRI data? What is that?
So
Okay.
so this study looked at, you know, Fitbit actigraphy, and then they corresponded that with fMRI data. So around the time that they were doing the actigraphy, the 2 weeks of actigraphy, they were measuring kids' like functional brain capacity, right? And we'll talk a little bit more about that later. So essentially, these are patients who did not get the fMRI done, right? So that excluded about nearly 1,000, which, you know, thinking about what could be why may have patients not gotten the fMRI, right? Could it be like transportation issues, or like parents are working, you know, they're not able to take off work. So there may be
okay. I was talking more about the outliers for fMRI data, the one that had that N of 87 with greater than 5 standard deviations.
Oh,
is it
okay.
that the fMRI data was just so like far out from the average or the large majority of the patients that they felt it was not?
Yeah, I'm thinking that that greater than 5 standard deviation. So I'll show you the fMRI data.
Okay.
they measure, I don't, you know, this is another weakness in my opinion to this test, to the study. I mean, is that it's very esoteric, and I've read this a bajillion times, and I still have a hard time understanding the actual data. But yes, my understanding is, you know, they plot like duration of mean sleep duration based on the actigraphy, right? And then the y-axis is like essentially what they found on the fMRI data, like the ability to make connections, intraconnections and interconnections between different like neuronal groups that relate to like executive functioning, cognition. And so I think that this outlier is like, if they're beyond the 5 standard deviations of whatever that measure is for connectivity. You see what I'm saying?
Yeah.
And
thank
Evie,
you.
some of the other outliers on there too that they excluded, it was like frame-wise displacement. So if there was significant motion artifact on the MRI, I think those
Got
are
it. Okay.
Okay. And then we're gonna scroll down. Can't scroll on this. Okay, so this is our demographic table. Does anyone want to tell me what they see? Just let me yeah, does anyone want to tell me what they see or any potential biases in these demographics that
I mean, off the bat, just going down, I think ethnic sample size may be important here. You know, it's looks like a 60 to around 60% was white, less than 10% Black or African American, and then 20% Hispanic and 10% about Asian. So it seems to be skewed largely towards a white population or white children in this case. So that might be like, you know, one of the things. Not saying that that necessarily is significantly going to affect the data. It's hard to say without going into the data. Obviously, we haven't got there yet, but just something to keep note of.
Yeah, for sure.
I would say though that like compared to most big studies, the distribution's better than most, I would say.
Yeah.
It usually does skew very much in the white category.
Yeah.
So that's something. But you're right. Like in these, ideally.
For sure. Yeah, no, I agree with what we both said. You know, every study's gonna have its problems, right? So anyone else want to say what they think about the data?
I think I was actually gonna you highlighted. I think that's actually very good that they included that, because that's something I was thinking about, was the socioeconomic status of the kids. And they seem to have a pretty healthy distribution of based on income, household income. So I think they did a good job of making sure they got a representative sample size to include multiple I guess ranges in terms of socioeconomic status for the kids.
You know, I actually would disagree with you, Evie. I feel like it's biased towards rich people, right? Because like if you think about how much of the population is above 100, that makes 100 above 100,000, right? It's very low, but nearly half of the kids were above 100,000. So I thought that there was bias towards rich people.
I would agree. It was
yeah. Okay. And then I thought this of all when I read this, I was like shocked actually by this. If you look at like the Fitbit reported hours of sleep, like it's like 7.5 hours, like is like the major distribution here. Like my goodness, I had no idea. And also, like, what do you guys think of this data? Like in terms of, do you think the Fitbit tends to overestimate, underestimate total sleep time? What are your guys' thoughts?
just looking at those charts, it's probably a pretty fair estimation. I mean, that's a pretty nice bell curve.
Distribution, yeah.
I mean, are you surprised that they're getting that much sleep?
No, like that little sleep. I guess maybe I'm naive. I don't have teenagers. I guess I don't remember how much sleep I got when I was a teenager.
So Brad, they gave like minimal details about how they cleaned that Fitbit data.
Yeah.
So, I mean, you know, the relationships are still the relationships that they found statistically, but I'm just like a little dubious of what the actual sleep duration was. Because we know that these devices, like depending on how they were using those timestamps of when they got in and out of bed, like sometimes they'll miss entire windows of attempted sleep, or sometimes they'll capture windows when people are so for this population, I don't expect a lot of them were cell phone users. But if you don't, if the time in bed is not correctly established, there can be huge spurious findings with these. And I'm actually surprised it got through into sleep without more details on how they clean their Fitbit data.
Yeah.
It's just something to keep an
Absolutely.
eye on in these studies, particularly how they're designating the time in bed period.
Yeah, absolutely. And I think my watch will go off, right? If I'm sitting down, it'll say like it'll go into sleep mode when I'm like watching TV. So it's like
Exactly right. That's exactly what we found. Yeah.
So yes, I completely take this with a grain of salt, right? The sleep duration. Okay. Okay. So now let's go into or does anyone have any other thoughts up to this point about just like the study design in general? The biases that may have come up. Okay. So overall, it's a robust study, a lot of data, but may have some biases, but like all studies have biases, right? And so kind of maybe goes along the lines of in terms of demographics, what other studies may be, but certainly the Fitbit data is questionable. Okay. And so this is maybe someone with more of a neurology background can help explain this. Like I said, I read this a bajillion times and I still have a hard time like understanding exactly what any of this stuff means. But I'll try my best. So basically what they found is that sleep duration has a positive association with 5 network domains, which I wrote them down here. I can't remember. Emotional regulation, memory consolidation, and I think cognition and attention. So basically, more sleep led to stronger connectivity between these networks, which is a good thing, is my understanding. That's maybe it'll help if I pull up the brain diagram that they have. Let me, is it at the top? Okay. And this is separating it into girls and the boys, right? So essentially what this is showing is it's showing these, I don't know, like groups of neurons, I think, that are like with the fMRI show connectivity to have a relationship when they light up, detecting whatever they detect on the fMRI, and connectivity between different groups. And so in girls, you can see here that the orange shows that there's greater variation between the interconnectivity between these neural groups. And in boys, there's less variation. I don't know if anyone wants to, I'm not too familiar with fMRI. I gave it, I tried to read about it, but it's challenging for me to understand. So I thought, like for me, that may have been a limitation for the study. Like it's very challenging to understand what the results mean. But in general, I think what they're getting at is that more sleep is better.
That's what I got out of it too.
That's what you got out of it too? Okay, great. But that being said, it's more of like we've always assumed these things, right? We've always assumed that better sleep is better for cognition, emotional regulation, cognition, executive functioning. But I think this is like one of the first studies like this, that is trying to put objective measures to these things, right? And so, you know, at least you're seeing like this is like, you know, big cohort of kids that where you have positive correlation between correlation between mean center duration and these connectivities on fMRI. I still don't quite understand like the differences between boys and girls either, or why like why they delineated that. But I also thought it was very interesting because they also found negative associations, right? So you have mean centered sleep duration has a decrease in these connectivities. So I was like, I think maybe here it's like talking about this decreased connectivity may be less like hyper arousals or like less, noise in the brain, I guess, that may that is associated with better sleep. I'm not sure, to be honest.
So, I was looking at that and I was thinking to myself, from my understanding, like, I'm not a neurologist, so I defer to the neurologist in the meeting. Like, my understanding was fMRI is an idea of brain activity and how different areas in the brain, when they're active, how they may connect with each other. That's just as much of an understanding I have of fMRI, but I do understand it's not as simple as more connections is better. It's also dependent on what areas of the brain are connected to each other. So I think that may be why you're seeing negative associations, but that doesn't necessarily mean it's a bad thing.
Right.
And so that was my takeaway from that, is just because there's a negative association between sleep duration and connection to certain parts of the brain, certain parts of the brain, between certain parts of the brain, that might not necessarily be a bad thing. That might be showing that, well, or let's say you sleep less and you have more connections between certain parts of the brain. Well, that may be interpreted as not necessarily a bad thing. It may be that, like, well, that's what happens, where these connections that you don't normally expect to see them, you start seeing them, and then it's not necessarily a positive thing. This is, like I said, where my understanding of fMRI and the, like, what the data behind fMRI means in this situation lacks, like, any sort of concrete, I don't know, interpretation here. Like, I would definitely defer somebody in the ECG realm.
Yeah. And I think, you bring up a good point. Like, it's like they're showing these like correlations, right? But what do they really mean, right? It's just like a finding on fMRI, right? And so they did break it down. This is where they do the NIH toolbox, right? The cognition battery. So it's like a, it's a research accredited back way of measuring certain brain things. And so this is where they found that they found that better sleep was associated with better cognition, cognitive functioning, emotional regulation, with greater sleep duration. So this was like another outcome that they tested, was based on the NIH toolbox performances.
Did they adjust for the billion correlations that they make at the same time?
Yes, I believe so. Like, even like in terms of like adjusting for like age, sex, race.
No, yes, they did do that. But what I'm asking is, like, if you, like you show in that table or these graphs, they're actually the table, I mean, more so, you've got a lot of p-values. So let's say you do tests to generate 1,000 p-values, right? And you're identifying p less than 0.05. Well, you're going to anticipate that at least 50 of the p-values are going to be positive and show you an association. And so Kathy just put something in the chat that she found that they did adjust. Let's see. Did they penalize?
Yeah,
you're
I was asking, did they?
Yeah, that's what I'm asking. Is do they, and penalize? It just means you have to be more conservative in your conclusion to avoid kind of automatically having 50 spurious associations.
Mhm.
And I think a lot of times in fMRI research, they do that, but I didn't find where they did that here, but I didn't know if that struck you or Louise or anybody else.
Yeah, I'm not sure. I don't know where.
Okay.
Is that what the like Benjamini-Hochberg procedure is? Is that what that does? I don't know, because they'd made mention of that too. For like
think they
false
still keep
discovery
the-
rate correction.
Yeah, they do mention that, but I think they still had the P as less than 0.5. So I get that they corrected for a false discovery rate, but then I don't understand why the P values are still less than 0.05. So I'd expect them to be different.
Oh, because they corrected the p-values. Yeah, okay. They did they
Corrected
correct.
them, but then they still say that less than 0.05 is regarded
Yeah.
as significant.
Yeah, but that probably means that they they started with a p-value of 0.001 and then put it through the correction and reported it as 0.05.
Okay. Yeah, I don't know what that method is, that Benjamin thing.
Yeah, I haven't used that. Okay, well, that's helpful.
Okay. See what's the next. Okay. So, does anyone, just before we kind of go into the end here, we have about 13 minutes left. Does anyone else want to bring up anything about the article that they thought, like a good, like overall general impressions? Like, does anyone want to speak up to say, like, do they think this is good, bad? I know, the actigraphy data is questionable, and there may be some biases limiting the interpretation, but
Well,
overall, at the 10 to 13 years old, the 7.5 hours of sleep is so low. And
yeah.
what are they basing this? The greater sleep is equal to that connectivity based on this low number of hours of sleep. You don't have a control of, somebody who is sleeping for their age group at least 9 to 10 hours of sleep with that age group, and having, doing this functional MRI with these toolbox connect, and how their connectivity is different from these group of kids.
Yeah,
And-
no, that's a great point.
for me, there's another red flag in the language that they use in interpreting their results. And the title is correct, right? Associated with blah blah blah. But they go to assuming or implying cause and effect pretty fast.
Yeah.
And that's always a red flag for me. And I'm been either an editor or associate editor or deputy editor with Sleep for many years. And I can only tell you that when I see that, it's a pretty big negative for a submitted manuscript. And especially if it's not easily correctable because it permeates the whole thinking of the manuscript, it's the best way to get rejected. So I don't know what happened in this review process that they didn't, they should correct that and call it out. And at minimum, ask people to be more circumspect about assuming causality.
Where are you seeing
So-
where are you seeing the assumptions of causality?
Oh, various places, like I saw it in the first paragraph of the discussion, I think, if I remember. I mean,
Let's look at their conclusion. Their conclusion looked okay.
All right, well,
I think
maybe
they're
not
looking at
so hard.
associations.
Okay. So just rounding it out. Does anyone want to tell, does this study change how you counsel families about adolescent sleep, or make you think of any future research opportunities? You know, one thing I thought about was, you know, I think with ADHD, right, in the past there was a study that showed that, you know, this actually may be sleep apnea, right? And so this was like before that there was known that ADHD was associated with sleep apnea. And so now it's like kind of common practice, right, to screen for ADHD in the pediatric sleep population, because that's a big indicator that they may have sleep apnea, they have attention problems. And so, you know, this study may argue that should we be screening more for like emotional dysregulation, right? Could that be related to, you know, sleep deprivation, or, you know, poor decision making? So does that mean, you know, is that related to sleep deprivation? And should we be paying more attention to that in our clinical practice, or I don't know. Overall, you know.
Question. I don't know if I missed this. You said they didn't screen out pediatric patients that had sleep apnea. They didn't look for it at all.
No, they did not.
Okay, so they didn't. Okay. And the other thing that I thought may be interesting as a future direction, and I don't know if I didn't look deep into the graphs that they displayed to see if this relationship was shown, was if the fMRI connectivity patterns that they noticed in sleep deprivation, so less than 8 hours in kids, I guess, were similar to those kids that slept longer than the recommended time. So what is the fMRI connectivity look like for kids that maybe are sleeping 11 hours a day? Is it similar to sleep deprivation? Is it different? That'd be something interesting to look at.
Yeah, no, you bring up a good point. They mentioned that in their limitation here. The U-shaped associations, I think is what you're talking about. The U, you know, the two extremes, right? They only looked at the sleep deprivation, and I think they only had one kid that was above 10 hours.
Yeah.
Which right here, excessive, which is normal to me, but no. That's a great point. Like I said, you know, I think this is a very robust study, a great attempt at trying to put data with things that we kind of already have been doing, and you know, that's
I guess kind of going back to your one question, does this change like your clinical practice? I don't think it changes the way that I would counsel. Right, because if we're just saying that we're trusting the Fitbit to be accurate in this, right? I would never tell somebody to get less than 8 hours of sleep, regardless of what the data shows.
Yeah, absolutely. I 100% agree with you. Yeah.
So I think it's like a good first step, right? In trying to figure this out, but it does not change the way that.
Yeah, there are too many problems, I think, also with like the study, like the the Fitbit, and
I think like way in the future too, it would be interesting to include some of the, you know, because they had mentioned that they excluded the severe neurologic conditions and like psychiatric conditions. So I think it would be interesting to see
the differences there.
But,
yeah, for sure.
Much farther down the road.
I think
I also
adding that, sorry, go ahead.
Sorry, I was just going to say, despite, you know, there's certainly a lot of critique and, you know, sort of negative things, I guess you could say, about the study, but, you know, to measure in almost 4,000 children objective measures of sleep, that's a big deal. You know, you certainly couldn't do that in a research project with a PSG unless you had a lot of money. So I think there are some, you know, some things just to acknowledge that's a big deal to do objective measures of sleep and fMRI, even though there are certainly, you know, challenges and and lots of things we could critique it for. I think it's still a pretty amazing undertaking, and it's given some objective data behind some of the things that we already know, right? That sleep is linked to all sorts of outcomes, and this can give potentially some objective data behind that, the mechanisms potentially.
And I don't think the Fitbit is a problem in and of itself, right? Like, I love these devices for sleep research. The problem is when people don't appropriately handle the data, or they don't provide details about how they handled the data. There can be a lot of spurious findings. So, I mean, maybe they handled it correctly. Maybe they were recalculating everything based on those timestamp bedtime and rise time, and it was verified with like the parents, but it's pretty, like, there's no details in their methods. And I just always get nervous about there being like, tens and hundreds of thousands of subjects with data like this that we're letting go through into, these high tier sleep journals. So it's really important to look for that in the methods, because they can be used appropriately. You just have to make sure that the data is handled well.
I think on the flip side, looking at this a different way, I don't know that this would compel me to, encourage people to get trackers for their adolescents as part of what they're doing at home either. I think sometimes patients want to know if this will influence kind of our management overall, and I don't think that I would be so compelled, to encourage people to track their kids' sleep.
I completely agree. All right, guys. Well, any last thoughts? But anyway, well, thank you guys so much for your participation, and I hope you go about your day taking a deep breath, and have a great day.
That was great, Brad. Thank you. Great discussion.
Great discussion.
Yes,
Thank you.
thank you so much.
Good job, Brad.
Thanks,
Bye-bye.
Louise.
Bye.
Thanks, Brad.